So we start on the second half of the morning with Pierre-Jean Coringet. He's director of the Channel Receptor Unit at Pasteur Institute, and he'll tell us about allosteric transition of the pentameric ligand-gated ion channel.
So, hello everybody, and it's an honor to be here and to present my latest work. So, I've been working in the field for several years now. Actually, I started in '93 when I went as a researcher in Jean-Pierre's laboratory. And I've been working for 10 years in Jean-Pierre before doing my own group. And I read the paper of Palmer years after years, and it has been really a great source of inspiration for my work. And actually, I will show you some fluorescent work in my group that are inspired partly by Palmer's group. So thank you, Palmer, for the invitation. So I want to speak about what we are doing in the lab. We are working on a bacterial receptor. So the title is a bit weird. It's not the pentameric ligand-gated ion channel. It's a bacterial homologue of this family of very important receptors. So I don't need to introduce those receptors. They've been introduced by Stephen and by Ryan. So I am interested in the same kind of topics. How does an electrophysiological recording translate into structural at high resolution? So basically, the interest is in allostery. So this is a dose-response curve of a nicotinic receptor. So at low concentration, when you apply acetylcholine, you have a transition from resting to active state that you can see here at low concentration. And we know, and I will speak about you more or less, and from Stephen's talk, that there are some intermediates in the course of this activation pathway. And when you activate at higher concentration, you have new states coming, which are desensitization states. And this is, I would say, for us, the minimal allosteric model that describes the function of these receptors. So the structural biology of the field is growing very fastly at the moment, and this is a snapshot of the main structure that have been solved. So the first full-length structure of the family were solved thanks to the discovery of receptors from bacteria, which are simple in structure, which are smaller, and which are more easy for structural biology. So two receptors were known, ELIC from Erewina chrysanthemi, and GLIC, the receptor I will talk about. So after that breakthrough was the structure of the glucial by...
Yeah. They're videotaping here, so they wanted us to point at this.
Okay, screen. That's fine. So the breakthrough was the first structure of the eukaryotic receptor by Ryan in Guo's lab, which was followed by several structures, the GABA beta-3, glycine alpha-3, 5-HT3, and the last receptor, the glycine receptor alpha-1 by cryo-electron microscopy. So you see that we have more and more structural data. But still we are working on the GLIC channel, which is a prototype for the whole family and is very favorable. So GLIC has many advantages when doing structural biology. I will show you that. So GLIC, so it's a channel from bacteria, Globacter violaceus, from the cyanobacteria, and it naturally lacks a cytoplasmic domain. So this is a full-length structure of GLIC. And we show a few years ago that GLIC is a proton-gated ion channel. This is a dose-response curve. It's activated starting at pH 6, and maximal activation is found at pH 4.5. So we made several cameras based on GLIC. I show you one of those cameras, extracellular domain of GLIC and transmembrane domain of the glycine receptor. Actually, we recently solved the structure. But just to show that this camera is also a proton-gated ion channel, we show that we don't know where proton act to activate the channel, but definitely it acts at the extracellular domain. We also identified some molecule which modulate the function of GLIC, and in particular caffeic acid is a negative allosteric modulator, and we found that it's a modulator which binds at the level of the orthosteric site. So although GLIC is not a neurotransmitter-gated channel, there is a pocket for allosteric modulation on the protein. So, advantage of GLIC. GLIC is really favorable for crystallography, and these are several crystal packing which were generated by my collaborator Marc Delarue in the Pasteur Institute. And among all the receptors of the family, GLIC has been solved at the highest resolution, which is 2.4 angstrom from this C2 form. So just to summarize what we did at the structural biology with Marc Delarue, we first solved the GLIC structure at acidic pH, and this is the structure that was solved. So you can recognize the pentameric organization. Each monomer is colored differently. Extracellular domain folded as a beta sandwich. Transmembrane domain folded as alpha helix, 4 alpha helix per subunit. And the quality of the density map was good, and the crystallographer could construct some molecules which don't belong to the protein, in particular lipids. So they are shown here in spheres, blue spheres. You can see the lipids which are co-purified with the protein after extraction from E. coli. Another surprise also is that you can find some detergent molecule in the structure, in particular a bundle of 4 DDM molecules that plug completely the pore. So this was the first form to be solved and to be published in 2009. And we think we have an open channel conformation. So there are a lot of arguments, but I will just give you the 2 main arguments. The first one, this is an ion permeation pathway. This is an ion channel itself. It has a funnel shape with a large diameter outward, a constriction inward, and this organization is consistent with all the biochemical data that have been collected on the open channel conformation. Another argument is that this structure was solved at pH 4, and at pH 4.6 you have the maximal activation by electrophysiology. So the fact that this structure is open is still a controversy, in particular because the last paper on the glycine receptor showed that you have the same kind of organization, but expansion at the cytoplasmic side. So there is still controversy, but according to our molecular dynamic simulation in collaboration with Mark Baden, we pulled ion through the pore, and the simulation is consistent with the permeant conformation with no drastic energy barrier for the ion to cross the channel, with at the cytoplasmic border of the channel partial dehydration of the ion to pass through, consistent with a selectivity filter at this level. So we, after work hard to get a closed channel conformation of GLIC, obviously to have an activation pathway of the molecule, and this was performed by Ludovic Sauguet in Marc Delarue's group. So it's a hard work, and the structure that was published was at 4.5 angstrom resolution after thousands of crystal paths, so this is the best resolution they could have. The asymmetric unit is huge, with 4 pentamer in the asymmetric unit. But despite the moderate resolution, the electron density map is nice. You can see that you can nicely build the helices and the sheet of the extracellular domain, so you can nicely build the peptide backbone. So according to these 2 structures, one structure at pH 7, which corresponds to the resting state, one structure at pH 4, you can decipher mechanisms for activation of GLIC. And these mechanisms involve first a global reorganization of the protein, a twist of the whole protein, and also what we call a blooming motion, is that at pH 7 the extracellular domain are detached from each other, and at acidic pH they come closer, like an unblooming of a flower. Another key feature of this transition is, of course, channel opening. So in the resting state, the channel is closed by 3 rings of hydrophobic residues, which are shown here. And upon opening, you have a tilt of the N2 segment to open the upper part of the pore, which is the activation gate. So just to show you this transition, I show you a movie starting from the resting to the active state. So this is just a morphing, just to visualize the activation transition. So you can see the extracellular end blooming motion from the side and upper view. You see that the subunit come close together and the interface narrows down during activation. So this is consistent. If you would have neurotransmitter binding that, you would have increased affinity for the neurotransmitter, and actually this was shown also by Ryan Heath in the glycerol structure. You have the same kind of transition. So then going to the interface between the ECD and the TND, you have a local reorganization and a major rotation of the M2, M3 loop, which is shown here in red. You can see here in upper view, a major rotation. So this seems to me the major sensor, so the M2, M3 loop is at it shows the interface between subunits, so it sends the reorganization, moves, and in the movement, it will open the upper part of the pore to open the gate. So this is the speculative model we can build up from these two X-ray crystallographic structures. So my main concern here is to bridge the gap between electrophysiology and X-ray crystallography, because what we are really interested in is what happens to the membrane-inserted protein and the electrophysiological response. But to do an X-ray structure, you have to extract the protein from the membrane and solubilize it in detergent. And then you have to pack the protein in 3 dimensions, and this is what's shown for GLIC. And so you can easily see that this is non-physiological conditions, and how to bridge the gap between this structure and the membrane-inserted protein. For example, in GLIC, we have several examples, paradoxical example. We have the open form. We bind the open form with negative allosteric modulators, such as general anesthetics, and the receptor stays in the open form. In other words, the crystal packing selects some conformation of the protein. So to bridge the gap between this and this, we have to develop alternative approach, complementary approach to X-ray crystallography. And what we chose in the lab was to develop some fluorescence labeling to follow the reorganization of the allosteric transition at the membrane. So fluorescence has several advantages. So you can make measurement on membrane-inserted receptor, on freely moving protein outside from a crystal. You can make measurement in physiological condition, not in frozen and with a physiological buffer. You can make some detailed pharmacological studies, and last but not least, you can do some real-time kinetics to follow the reorganization during time. So the main approach for such purpose is usually to do FRET, fluorescence energy transfer, that maybe most of you are familiar with. But FRET has several limitations. In particular, the region where you have change in FRET are rather high, and mean usually the R0 is in the range of 3 to 5 nanometers. In other words, you can monitor with FRET regions which are far apart in the protein. And the purpose of the study here is to look at the local organization of the protein all along the gating pathway. So this is why we chose an alternative approach, which was developed by Mansour and Farenz in 2002. It's a biochemistry paper, which is a tryptophan-induced quenching method. So the principle of the method is quite simple. You introduce a cysteine on the protein and you graft it with Byman, which is a very small fluorophore. So the product is slightly larger than tryptophan, so you make minimal perturbation of the structure of the protein. And you can introduce quencher residues such as tryptophan and tyrosine, which quench the fluorescence only when the two side chains are in contact. So this is known as Dexter quenching. So if the two residues are far apart, you don't have quenching, you have full fluorescence, and if you have collisional, you decrease the fluorescence, and then you have a qualitative sensor of the local conformation of the two positions. So Byman is grafted through cysteine, like using classical chemistry, and in the original paper they made the proof of concept. So they introduce a tryptophan on lysosome, and they put BIMAN on several positions and monitor the fluorescence of BIMAN in the presence or absence of tryptophan. And you can appreciate that when the distance is high, you have significant quenching. But when you are very close to the tryptophan, you have a lot of quenching. So the technique is really monitor the distance between the two positions. So let's go back to GLIC. So we want to monitor the reorganization all along the gating pathway. And so we selected a key position, selected for the accessibility to reaction, and also for the movement from the movie on GLIC. So we first selected the loop B, which is at the top of the ECD, this aspartate, and this arginine, which is actually in the orthosteric binding site. Then at the bottom of the ECD, loop 2, which is a position which does protrude at the interface between the ECD and the TND. Then this proline, which is in the M2, M3 loop, which has a typical revolving motion during activation. And finally, a position at the top of the M2 segment to monitor channel opening. So we introduce BIMAN on each of these positions and make a mutational analysis. So first, the two positions at the top of the ECD. So we introduce first BIMAN at this position, and then we mapped out tryptophan residues to find a good pair of quencher. So you can find that some tryptophan were not very interesting. So this is the BIMAN alone. So this is a dose response curve in cuvette. So we put the protein purified in detergent in cuvette, and we monitor the fluorescence, and we progressively change the pH. So with the BIMAN alone, there is no change in fluorescence. And when we introduce this tryptophan at this position, we have a very nice sensor of the distance. The signal is high here, and you have quenching, which means that the two positions goes near together during a pH drop. The same thing for this position, 133. We find a tryptophan which has the same position. And so these experiments show that we are at both interfaces of the subunit. So you can see the crude data. You have a very important signal. And it shows that during acidification, those two positions goes together, and those two positions goes near together, which fits very well the unblooming motion shown by X-ray crystallography. So then we are moving to the ECD-TMD interface, and the things are a bit more complicated. So first, this residue 33, there is a natural tryptophan nearby. And so actually, when you put a BIMAN at this position, you already have a strong effect of the pH on the fluorescence. And actually, when you remove the endogenous tryptophan, you remove this difference. So here we monitor that both positions are going close together during acidification. Next, the proline 250. Actually, the proline 250 is in a region where there is a lot of tyrosine and tryptophan, so you have a lot of quencher around. So at this position, we mutated back the tryptophan and tyrosine into phenylalanine to find partners. We mutated a lot of one, but one got the same position, so this is the result. This is the BIMAN alone. You have an effect upon acidification, and when you remove this tyrosine, you have no more effect. It means that during acidification, the two residues are going apart in the structure. And finally, at the top of M2, this glutamate ring, we didn't find pairs, but there is an effect of fluorescence. So here we have a qualitative sensor of the local conformation, but we cannot assign it to a particular pair of residues. So altogether, this fluorescent data fits well the model from X-ray crystallography. So this is the same movie, so going from pH 7 to pH 4. And you can see that the balls are the BIMAN location, and in stick you have the quenchers. And you can see that both positions are going closer together. These positions are going closer together, and if you go in upper view, this proline residues is going away from the quencher, and all these features were shown by fluorescence. In other words, the fluorescent residue, the fluorescent data are consistent with the activation model from the X-ray crystallography. So next is the allosteric organization conserved in liposome. So we produce the GLIC and we reconstitute the GLIC protein into liposomes and measure the fluorescence by the same approach. And actually, we found that the protein motion are conserved in liposome, especially in the extracellular domain. So these are the pattern in gray in DDM, and in black in liposome. You can see the pattern are identical. In other words, the allosteric transconformation of the protein is completely independent, insensitive of the environment of the membrane at this level. At the TMD-LCD interface, things are a bit more complicated. You can see some differences in the intensity of fluorescence at several positions, but overall we have the same trends for the fluorescent change. In particular for beam 33, you have a decrease in fluorescence in liposome. You see also a decrease in fluorescence in detergent. And for beam 250, you have the same trends, and also this way. So in other words, you have a strange change, maybe due to the environment of the transmembrane domain, but the local motion seems to be conserved. And this is a validation of the X-ray model that the allosteric motion are conserved in lipidic environment. So what about the activation transition? So we wanted to compare this fluorescent data with the dose response curve by electrophysiology. So we express the various constructs in the oocyte. We label them with Byman, and we record the classical dose response curve for activation. Sorry, the slide is a bit complicated because all of this is not published, but I will be fast on this. So this is, for example, this position at the top of the ECD. In black, you have the dose response curve for activation, which is channel opening. And here you have the dose response curve for fluorescence change. And this is the same for all the positions. Overall, you see that you have a match which is not bad. I mean, you have the fluorescent change at almost the same concentration as the activation. But you still have a difference when you go at the start of the dose response curve. You can see that the fluorescent signal is already there, and you still have no channel opening. For all positions, you have a change. And so the fluorescent change are more sensitive to proton than channel activation. This is valid for all positions except the position at the top of M2. And so we can say that for this position, the movement occurs at lower proton concentration than activation, and it's indicate that you may have intermediate in the course of activation, maybe asymmetry, or maybe intermediate conformation, but it's not that clear at this level. So next we move to stop flow experiments. So with the disfluorescent sensor of the conformation, we can monitor the change in conformation in real time. So we move to stop flow experiments. So you put the GLIC reconstituted in liposome, buffer, and then you mixed and you change the proton concentration, and then you make some real-time recording. So just to remind you, maybe you don't know that GLIC is a slow channel. So this is data by outside-out patch electrophysiology made in Tchaikovsky laboratory. They show that, for example, the GABA receptor is a fast activating channel with an inactivation in the millisecond time range. In contrast, GLIC is rather slow with a tau of 150 milliseconds, so it's a rather slow channel. So we move to stop flow experiments, and here are the results, which were rather surprising to us. So this is the traces of the Byman at the top of the ECD with his quencher. So you have here at pH 7, you have no effect, and then at pH 8, sorry, and then you change progressively the pH to acidic, and you can see the drop in fluorescence. But the big surprise was that actually the drop of fluorescence occurred in the bed volume of the apparatus. So this is an enlargement. So this is 20 seconds. You enlarge to 100 milliseconds. You can see that just after the 2 milliseconds of the dead time, the drop in signal is already occurred. In other words, the change in conformation between these two positions occur less than 2 milliseconds. So this was found for this position, but this was found for almost all positions. On beam 33, we have exactly the same pattern, almost. Also on beam 250, we have a similar pattern, very fast. And the only notable exception is at the top of the M2 segment. So we have a very fast signal, but you have after a slower signal, which occurred at pH 4 and pH 5, with time constant that would fit with the activation of GLIC. So this could contribute to the activation. And so to put all this data into a scheme, what we show, so this is a scheme of a GLIC with the extracellular domain, transform brain domain, M2. These are the position in loop B. At the bottom of the ECD, this proline in the M2, M3 loop, and the position at the top of the M2. What we show is that upon pH drop, you have so the unblooming motion, and you have a lateral motion of the M2, M3 segment, and this occurs very rapidly in the 2 millisecond range. So you go to what we call a pre-activated state, where most of the allosteric transition has occurred, but where the channel is still closed. And then you have a slow transition, which is in the 50 to 100 milliseconds, with a local motion of the upper part of M2 to open the pore. So it's interesting because this pre-activated state resemble in some way one state we already captured by X-ray crystallography, which we call the locally closed state. So this was a work published in 2012. Actually, in this work, we crystallize a cysteine bridge version of GLIC. So this is when the cysteine are reduced, you have the classical open state. But when you bridge the cysteine together, you capture a new state where the upper part of the channel is closed, but the rest is in the active conformation. And we call it the locally closed conformation. Actually, this conformation was observed for a lot of mutation on GLIC. And we propose, so the pre-activated conformation would match partially the locally closed conformation. So this is a movie starting from pH 7, going to the locally closed and to pH 4. So you see the two steps for the activation this time. And you have first the unblooming, and then you have the revolving. So it doesn't match really, because from our fluorescent data, we predict that the extracellular domain and also the loop move before the opening of the channel. But this is a first hint to try to have an idea of the intermediate state. I have a few minutes. I have to conclude.
Just a couple of minutes.
I am almost done. But this is my last slide. Just to summarize all the data. So the X-ray structure from GLIC between pH 7 and pH 4 allow proposing an activation mechanism of the protein. The fluorescent tryptophan-induced quenching method support the X-ray mechanisms both in detergent and in lipids. And most interesting, the kinetic analysis show that activation involves a pre-activated state. And from the previous talk by Steven Sine, we can speculate that this pre-activated state might contribute maybe to a priming state, or maybe to a flip state, as proposed by Colquhoun and Civilotti. And I just conclude about the author. So the key collaboration with Marc Delarue for X-ray crystallography, with Marc Baden for molecular dynamics, and all the people from my lab who contributed more or less to the work. And mainly I want to focus Anaïs Meni, who made most of the work on fluorescence, a work which is not yet published, but which is in the process of publication. Thank you for your attention.
One or two questions?
Pierre-Jean,
Yeah.
you
seemed to show that in fluorescence that both the detergent micelles and the phospholipid vesicles gave similar results.
Yes, that's true.
In the crystal structure, you mentioned the dodecyl maltoside could be seen. Is there any residual phospholipid in the crystal structure from its original isolation, or is it all replaced by detergent?
No. There are 3 phospholipids per subunit, which are co-purified with the protein. So we can see some phospholipids which are still there. Yeah. Yes.
So they're a different category than those replaceable by the detergent, perhaps.
So we don't know the nature of this phospholipid because we don't see the polar head, but we can see nicely the density of the 2 alkyl chains. We don't have lipids during purification, so they belong to E. coli. They were co-purified, so there is a tight binding of these lipids on the protein. Yes.
One more question? Yes.
Did you try the? So did you test the? I believe it was the glycine receptor that you swapped the transmembrane domain.
Yeah.
Did you test the glycine receptor for activation by protons, or is that no?
No, it's not activated by proton glycine receptor. It is modulated. I mean, most receptors are modulated by protons, but it's not activated. That's for sure.
That one in particular is not.
No.
And then with the dehydration, do you think that are there any molecules nearby? To give you an example, in the potassium channel, there's molecules that are thought to help with dehydration. Do you see those? And then do you think that that contributes to rectification? Because if in one direction there's different residues on the different sides when you have to dehydrate.
So for dehydration, we have serine, threonine, glutamate. So these are the side chains are there to help for dehydration, like in the sodium channel selectivity filter. So it makes a lot of sense. For rectification, I think glyc is not rectifying. And it's a very good question. I think nobody understood rectification on these receptors. That's a good, I don't know.
All right, we should move on. Thank you, Pierre-Jean. So our next speaker is Scott Hansen, Assistant Professor in the Department of Molecular Therapeutics and Neuroscience at the Scripps Research Institute, just across the street.
Okay, so I'm actually at the Florida campus. A lot of people don't know that Scripps built a separate campus in Florida, mostly for drug discovery. And it's an honor to be here, and I'm very grateful to Tracy and all those that organized this symposium. I picked out a few pictures of Palmer over the years. They're definitely fond memories. I was a graduate student between 2001 and 2006. 2005 was a very good year for us. It's when we published on the acetylcholine binding protein structures. And this is Palmer. We didn't know it was his birthday, and we were out to dinner and ran across the street and found some candles and sang him happy birthday in Spain. This is some events Todd Talley used to say when the cat goes away, the mice come out to play. We'd head out on the beach when Palmer went out of town and have a barbecue. And this is Pascal. We were working on the binding of the paper where they described cobra toxin bound to the acetylcholine receptor. And then this was the first and only mustache contest in the Taylor lab. And it was a week-long period where everybody tried to grow a mustache, and then a month-long period where we would. And one of the rules was you had to shave the chin had to be shaved, and we almost got Palmer to shave his beard. We were this close. And rumor was, and I don't know if somebody was speculating that Sue put the kibosh on it. It was like, no, you're not. Is that true? Or no? It never got discussed.
Much earlier it happened. It never happened again. again.
So anyway, I took the liberty of seeing what Palmer would look like with a handlebar mustache, and did a little bit of Photoshop and chopped out the beard. And I think he looks great with a handlebar mustache. I think it would have been fine. But in reality, as much as the science was stimulating, it was just a wonderful place to interact with people, and some of those people are still some of my best friends. So my talk today is going to be kind of in the same theme as what Steve talked about, trying to relate how Palmer influenced my career, and hopefully it makes sense putting it together that way. And the last part is really a project that we're working in my lab on, the mechanism of mechanosensation. Hopefully I get to that. It probably won't be a lot of my talk. So when I came into the lab, this is the question Palmer was asking. What are the molecular determinants of allosteric gating? And people have talked a lot about that. And I feel like one of the main definitions at that time, and that had from the very first when Palmer started working on the nicotinic receptor, was the idea of what is a ligand and ligand binding. And I've just defined it here as the reversible, specific, and dose-dependent binding of a substance to a protein to form a multi-state complex. And these were some of the predecessors to me. Steve, for example, and David Johnson was an early group. There was a middle group in the late 80s and 90s doing fascinating work on toxins binding. This was selectivity for a mongoose. And then Steve Keller and Elizabeth, this is an era of mutagenesis where they started mutating the receptor and doing mutant cycloanalysis with Brian Moles. And then, as was said before with Ryan, when I came into the lab, I felt very fortunate to be at a time when we started doing atomic structures. And really, I think it's important to point out, not just because I was part of it, but because I think this was some of the earliest motions of a protein in response to a ligand binding. And you know, there's been lots of stuff done since then, but at this time, really, there weren't any. There was no examples of how to see how a protein would move, and I felt privileged to see this very early on. Like this is how ligands induce conformational change. And there's a site of the ligand coming in and binding. And you'll see why I mention this later when I get into some of the later stuff in my lab. I'm very unconventional, as Ryan said. I just decided, let's go try to do something. And when I started my own lab, I have been continued in that theme of being unconventional, or like what Steve said, just do the experiment that answers the question. And you'll see that as I, hopefully today, as I describe the questions we're asking and what our hypothesis is. So my main question is, how does a cell membrane sense and transduce force? It causes pain. It also is responsible for touch. And another question we're asking, which I won't directly address today, but is related, is what are the molecules that can control conscious thought? So Roger will probably talk to you later about the molecules that control long-term memory, but before those long-term memories are ever formed, they have to be somewhere. There's molecules, as you're thinking right now, that have to be storing transiently those conscious memories. And we have a hypothesis. It's just a hypothesis at this point, and I don't know if it'll become controversial. This is the first time we're really presenting on it. But we have this idea that anionic signaling lipids set cell excitability through lipid-gated ion channels. So that inside the cell, this isn't synapse communication, this is within intracellularly, that the cell has to decide what is its excitable state, what is it sitting at. And we think that the molecules, if we could peer into the cell and see which ones are controlling that, are anionic signaling lipids. And that mechanical force is generating anionic lipids and signaling, giving rise to pain or touch, and that anesthetics block the generation of anionic signaling lipids. And through this block excitability, and that same excitability that causes pain, and that same excitability in mechanosensation. And you may be asking, well, what is a lipid-gated ion channel? And part of my objective today is to explain what we mean by a lipid-gated ion channel, and also I'll briefly describe fluorescent binding assay for ion channels and drug discovery, and then hopefully, if there's time, talk about a physiological example of the role of lipid signaling to ion channels and how that works in mechanosensation. So this began for me in this lipid-gated ion channel theory began in Rod McKinnon's lab. I was working on a channel that needed this molecule PIP2. It's called PIP2 because it had these two phosphates right here, 4' and 5' positions. And over time, the lipid, when it's depleted, this is current, the current decreases and the channel shuts off. And we can add back PIP2 and dose-dependently activate the channel. And if you remember the definition of a ligand, it's dose-dependently and specifically binds a channel. And of course, the McKinnon lab, is interested in structures of channels. And this is a tetrameric channel similar to the sodium channel that was shown earlier. And when we saw the structure with PIP2, it was bound in the transmembrane domain. And since having worked in Palmer's lab, I feel like I had a unique position to see what does it look like when a ligand binds to a channel. And I immediately, as soon as I saw this and saw the conformational changes that were going on, that this looks like a ligand, that the lipid binding to the channel was indistinguishable to me from a ligand. And so I'll show a movie too so you can compare the two. Hopefully, please. And this is, again, this is a lipid coming in and binding to the channel. We saw a large conformational change in the protein. And when you zoom in and look at the amino acids also were undergoing these conformational changes to intimately hold that lipid in place. And then we saw changes in loop domains that are controlled by the lipids. And then when it flips on its side, you'll see that the hydrophobic gate, this is looking down the channel, the hydrophobic gate opens and allows for the ions to flow through. And so at this point, we started thinking of this lipid as a ligand. And this is very different than the way everybody had been looking at PIP2 in the past. PIP2, this is the old model where people thought that it was just changing the curvature maybe through negative charge, or that through negative charge a domain might non-specifically stick to the membrane. And I was giving a talk at a Gordon conference about this, and Suzanne Jakowski, it was a lipid Gordon conference, and she thought that it would be helpful to write this up as a review, and which I did last year, and I actually think it's helpful too for ion channel people to think now about lipids as ligands. There's a lot of lipids in the membrane, and they're low abundant and highly specific. And so this is the model that we're proposing, that lipids diffuse laterally in the membrane, that they have an affinity for the channel, they come on and off, and that there's specificity that negates the channel. And these are a list of some channels and the specificity depending on where this phosphate is. If the phosphate's in the 5' versus the 3' position, you can see that many of these channels, when it's in the 5' position, it inhibits, or when it's in the 3' position, it inhibits, or in the 5' it activates. So the channel is sensing these signaling lipids and using it to control the cell. Again, remember our hypothesis that the anionic lipids are what are setting the membrane potential. And if you look at the list of channels that are controlled by lipids, many of them control the excitability of the cell. Some of them have obvious, like the calcium channel that you saw, which is voltage gated, have obvious other mechanisms to regulate them, but they still bind these signaling lipids. So then we started thinking about, well, if the signaling lipid is controlling excitability, then how is it organized? And how does the cell signal. So if we think about the system Palmer was using, it packaged acetylcholine and then it released acetylcholine, and that was the signaling mechanism. Now if we look intracellularly, we can see that PIP2 almost always, and this is not work done by my lab, but just out of the literature, we can see that PIP2 has an arachidonyl side chain, and that PIP3 almost always has a saturated side chain. And that's interesting because the membrane is heterogeneous. It has lipid rafts, which are cholesterol and saturated lipids, and the bulk of the membrane is polyunsaturated. And then there's also in 2012, Stefan Hell showed that PIP2 can form its own domains. So if PIP3 localizes to this membrane compartment and PIP2 localizes to this compartment, now we have separations of signals and we're primed for a signaling event. And the way we think signaling happens is an enzyme cleaves, just like an acetylcholine, cleaves the signal. It can be PLA2, it hydrolyzes the arachidonic acid, and then we think that would shift over, because it would become saturated, and shift over into a raft, or it could cut off the head group, and this is hypothetical, we don't know this, we haven't looked yet, but arachidonic acid through acetyltransferase could become saturated and then translocate into a lipid raft. And then we think endocytosis is the repackaging, the pulling out the signaling lipids, and these signaling lipids are then pulled into the ER where PIP2 is resynthesized and then it's shipped back out to the membrane to reset the membrane. And we think that this anionic cycle, anionic lipid is controlled, and that is what sets the membrane potential in a single cell. So this is our hypothesis, and then we want to then find evidence of this, and we have quite a few projects working on this. The anesthetic one, I don't have time to talk about, but we do have data showing that anesthetics control this process. But I think for today I wanted to talk about drug binding and ligand binding. It was a project that I started in Palmer's lab looking at the acetylcholine binding protein and trying to develop a high throughput mechanism for drug discovery of ion channels. And we thought that this lipid binding site would be an excellent place. One, we can show that this is really a real binding site, and then we can also use the binding site to do drug discovery. And so we have a fluorescent lipid probe. We can compete off. We're in a detergent micelle. I appreciate Jean Coringer's talk about the fact that in detergent, and also Ryan showing that things behave normally in detergent micelles. And so we picked a channel. We've done this with many channels now, different classes of channels. We picked TREC1, and it's a 2-pore domain potassium channel. It's mechanosensitive, it's anesthetic sensitive, and it's a target for chronic pain. And this is just showing the bound probe. We see an excellent signal. We can compete off that probe with PIP2 or phosphatidic acid, and then if we have phosphatidylcholine, we see no competition, and it independent of the acyl side chain. So this is showing there's nothing else present here. It's only a channel and showing that the lipids are ligands at high affinity. And I'm not showing the affinity, but these ligands are binding in the nanomolar range. And so then, of course, we want to do drug discovery. This assay is in 15 microliters. We can go down to 100 or 500 picomolar receptor, and now we have human receptors in a 15 microliter assay, and with a single prep we can do about 10 million assays. So now we can go through ultra high throughput, screen very large libraries for drug discovery. And we did a pilot round in 1280 well plate, or 384 well plate, 1280 compound library. This is just showing hits from that library. This is the Z-score. Anybody that does drug discovery, you know you need a Z-score. This is very good. We need something above 0.6 to go into ultra high throughput. These are just some hits that came out of that assay. Flufenazine was one of them. Flufenazine is a known antagonist of TREC1, and it's known not to inhibit TREC. So this is one we just looked to study to see is the assay working. This is our negative control. interestingly, we went and did a dose response curve to see if it was just low affinity or if it was direct competitive antagonist, and it was not directly competitive. It only partially competed off PIP2. And this theme has come up multiple times. This is showing that allosterically through the channel, it was affecting the binding of the agonist site of PIP2. And so in a detergent micelle, we are sensing both conformations of the protein. And now we have a drug assay for a conformational state in a purified detergent micelle. And I think that's very important. It's hard to do state-dependent assays and also to be able to do 10 million of those assays with a single prep. Of course, we went and confirmed that fluphenazine is an antagonist. This is electrophysiology expressed with TRAC expressed in HEK cells. Scripps, part of the reason I wanted to go to Scripps, and this comes from Palmer, is the fact that they are very serious about drug discovery. And Scripps, about 7 or 8 years ago, built several core facilities. One was a lead identification with pharmaceutical grade equipment. We have almost 1 million compound library, and we have a core of med chemists, so the chemists build the molecules, and we have a DMPK and a pharmacology core. So when we find a lead compound, we can go in and build a real molecule. And I want to highlight this for 2 reasons you'll see in a second, but Hugh Rosen was one of the first Scripps people to really develop a molecule, and he had the assay. The Florida translational team screened, found lead ID, then the synthetic chemist built the molecule, and it was ready for preclinical trials. And this was spun off into a company, Receptos, and just in September that company was sold for $7 billion. So I think that Scripps and I think UCSD and Palmer have seen this pathway of, in an academic setting, really building a drug. And the Scripps team, that core, built a molecule that had the proper pharmacokinetics to go into man, to literally go into clinical trials. And having that interaction of the professor at the institution and then the core facility to get the proper pharmacokinetics and build a drug, I think was unique, and that this was one of the first examples to show that this is feasible. The other reason I wanted to bring this up is because I talked to Hugh right after he sold his company, and it turns out the target was sphingosine 1-phosphate, and it's a lipid. And it was targeting, and I asked him about this, and everybody thought he was crazy that he was going to target a lipid binding site in a protein. And he did it anyway, and it's been very, very successful. And I think for us, that shows that these lipid binding sites, in fact, sphingosine 1-phosphate is almost identical to phosphatidic acid, and it's just the linkages of the acyl chains that differ from phosphatidic acids. And we think that these anion signaling lipids are going to be controlling a lot of very important processes in the cell. And that I'm happy that we're with the same team that developed a compound for a lipid binding, an anionic signaling lipid binding site, which is just a coincidence, right, that I happen to end up there. So in the last few minutes, I want to talk about how this relates physiologically. Like, how can we think about lipid binding in a cell, and how would it really affect us? So we're interested in mechanotransduction, and that's, force, touch. If you over-touch is pain, like you hit your finger, you feel that. And the whole entire mechanosensation field, all they've thought about is tension in a membrane. And force is just mass times acceleration. And there's a whole other component which has been completely ignored in the mechanosensation field, and that is the kinetic output. If you hit something, it moves. And if you have order, if you hold it stationary, then it still follows the rule. It just heats up. So if you hit a hammer on a sidewalk, the sidewalk has to warm up because you can't break this rule. Something has to accelerate when you apply a force. And then we go back to our lipid signaling molecule, where the molecules are organized laterally in the membrane. And then we asked, what happens when force is applied to the membrane? And can you disrupt this organization? For example, if a channel was in a raft, would you disrupt the raft and it leave the raft and then get exposed to PIP2 and open the channel? It turns out that something that's in the membrane is going to also experience any potential tension. So we wanted to avoid a protein that was in the membrane, just so people wouldn't argue about whether we were experiencing tension or whether it was literally lipid mixing. So we came up with this scheme to test first. We decided, well, what if there was an enzyme in a raft, a lipid modifying enzyme, and its substrate is outside of the raft, and then through mechanical force, the enzyme leaves the raft, finds its substrate, and then makes the signaling lipid that then would activate the channel. And so one of the ideal cases was phospholipase D. And the reason is phospholipase D makes phosphatidic acid from PC. It has two palmitylation sites, which palmitylation localizes a protein to a raft, so we at least expected it to be in a raft. And then also PIP2 is known to activate PLD. And remember our organization that PIP2 is outside of a raft. So this is very peculiar. Why is the molecule that's supposed to activate the enzyme outside of a raft when the enzyme itself is palmitylated and should be inside the raft? They shouldn't be near each other. So our first test was, well, let's go look. And in line with what Steve said earlier, you just answer the question in the way you can answer it. So we didn't do high-res imaging, but rafts are very small, and we started to learn super resolution imaging. This is SIM imaging, structured illumination. It goes down to about 100 nanometers. We labeled PLD with GFP, and then we labeled cholesterol-rich lipid rafts with cholera toxin. This is a toxin that's known to label those rafts. And then we just looked at colocalization. And indeed, they looked very well colocalized by the overlay right here. We wanted to confirm this colocalization with FRET. So if the molecule, if the raft labeled molecule is near PLD, then they should FRET, and indeed they do. And then when we add methyl beta cyclodextrin, which depletes cholesterol, we saw the colocalization disappear. And then we did a control with trunculin, which disrupts the cytoskeleton to show that it wasn't the cytoskeleton. So in the interest of time, we also did palm imaging, which goes down to 30 nanometers. I'm not going to show this because I'm running short on time. But we looked at PIP2 localization, and in the control, PIP2 was not, this is a different, I don't have to, well, maybe I'll explain it. So, do I have a few?
A couple of minutes.
Okay. So,
You're jeopardizing lunch, you see.
This palm imaging is high enough resolution that you don't get any colocalization because you can now resolve the individual molecules. So you have to use an algorithm to calculate the cluster distance, like how close they are together. And it turns out that when we look at PLD relative to PIP2, it's not near PIP2. This was relatively weak localization. And then when we added methyl beta cyclodextrin, we saw it increase. And of course, then we wanted to look at force, apply force, and see if PLD was activated by force. So did it leave the raft, and was it activated? And we could see that applying shear stress activated PLD. And then when we added methyl beta cyclodextrin, the force, and this is just a summary of it, force and methyl beta cyclodextrin was additive, and that's showing that PLD is indeed leaving the raft and activating it. And then we went to the channel and showed if the channel needs PLD for mechanical activation, then if we co-express TRAC with PLD, we see a strong signal. And then when we put in the mutant, then pressure no longer activated TRAC. And so we're showing the whole system now that PLD is localized to a raft. It needs PIP2. When you apply mechanical force, they mix, and then this PA is generated, and the PA produced from PLD was necessary for mechanical activation. And we think this is the first example of showing a kinetic-based mechanism for mechanosensation that's not based on tension. And this is just a summary. And I want to end by, this is my last slide. I want to end back talking about Palmer and the influence he had on my career. Palmer, we know, studied ion channels and enzymes, and it was this agonist that linked those two things. And as I was preparing this talk, I realized the agonist I'm looking at is phosphatidic acid within an ion channel, but I've also ended back looking at enzymes. And I didn't realize, how many people study both ion channels and enzymes? And I realized now the first person I hired was an enzymologist. And I don't think if I had worked for Palmer, I would have had the courage to just say, hey, let's go look at enzymes and how the enzymes are controlling channels. And we have a lot more work on anesthetics and how enzymes are involved in that field. And I think it's going to end up being a very important part of my career. And I do think it's based off of Palmer's example. This is my lab right now, and I'll just mention Cerrone, who did the drug discovery, Yul Young Park, who did the electrophysiology, and Nick Peterson, who did the mechanosensation work on PLD. And now I'm funded by those things.
Well, let's take time for one question. Anybody have a... Hey, Dennis?
Scott, in phospholipase A2, there's a PIP activator that's been known for a very long time, and it's a lysine cluster that it binds to. Is that true in PLD and in your receptor?
We haven't really worked on the structure of PLD. We've mostly used it as a tool. I don't know.
I thought your mutation was a lysine to arginine.
That's in the catalytic site. That's not in the PIP2 site.
Oh, okay.
And PIP2 isn't absolutely required for PLD activity, so there is some with that. It's just greatly enhanced when PIP2 is around. So there probably could be some activity of PLD in the raft. But we also think that GPCRs are probably transitioning lipases in and out of rafts to activate them. So they're in the very beginning, when there was a cell, there weren't multicellular sites, and there weren't organelles. So probably the first evolutionary organelle was actually the partitioning of a lipid, and then it was using that as a compartment. We think of it like a neurotransmitter vesicle within the membrane, and then proteins releasing the signaling content upon a stimulus. So, yeah.
Ryan, did you have a quick question?
Sure. The question's broadly about time scale. So the first part of the talk. So time scale. So is the time scale of KIR activation by PIP2 compatible with this involved lipids, hydrolysis, recycling pathway?
Yeah, so this is very interesting. Recently, the lipid field, so I've been going to lipid Gordon conferences and trying to understand this field. One thing that's really come out is the fact that the ER now is thought to actually interact directly with the plasma membrane. So there's protein interactions from ER. So we think they're very closely positioned. The other thing, and I didn't show this, was that we've looked at PIP2 organization in the cell. And we think that PIP2 is, we see striations. And we think that the fluidity of the microdomain combined with the proximal location of the ER is set up to have the right time frames. But we really don't have data to actually prove how fast things cycle. But something that's in very close proximity, I mean, it's just a matter of a vesicle. You can think about it as being as fast as neurotransmitter signaling. You have one membrane that's in close proximity to another, and you can repackage and send it back as fast as those events occur in synapse. So, since they're the same proximity, we're assuming they can happen at the same rate.
All right, thank you, Scott. So let me just introduce our last speaker of the morning, Rick Rotundo from Cell Biology and Anatomy at the University of Miami. And Rick's going to tell us about biogenesis and trafficking of acetylcholinesterase from basic cell biology to therapeutic applications. Rick.
Thank you very much, and certainly like to thank Palmer for the honor of being able to talk here today. I never worked in Palmer's lab, but I've known Palmer since I was a postdoc. I started as a postdoc in Doug Famro's lab in 1976, and I met Palmer a couple of years later, and certainly his work has had a major bearing on everything that I've done in my entire career. There's very little that I did in my lab that was not influenced directly or indirectly by his work. And certainly I discussed literally everything, almost everything that I've ever done with him at some point or another at meetings and other venues throughout the past 30-some years. So I'm very grateful for that. And I actually have a couple of things that I'll be presenting at the end that will be really fitting very well within the topic of this symposium on toxins and therapeutics. Anyway, so what I've been doing for the past 30-some years is, beginning with my postdoc, is studying how the acetylcholinesterase molecule itself ends up folding and oligomerizing and eventually being processed and ending up at a synapse. So this is a very cell biological question. This is.
Right now.
Huh?
Let's see. I gotta get the space bar right now.
Should I just
To go to the next one. I'm looking for the USB piece that I can't find. So when you want to go to the next one, you can either.
I'll just press that. Okay. So this is based on work not only from PalmerTaylor's lab, Jean Massouillet's lab, Israel Silman at the Weizmann Institute on the structure of the esterase. This is actually taken from one of Palmer's papers years ago, about 20-some years ago. Everyone knows that the acetylcholinesterase has multiple oligomeric forms. They're encoded by one gene, and the forms are assembled inside the cell in the endoplasmic reticulum, and they're associated, some of them, with different, one of two different non-catalytic subunits that people believe targets them to specific synapses in the cell. This is the types of synapses. This is the neuromuscular junction here, stained with fluorescent fasciculin, sites of nerve-muscle contact, epitomizing the kinds of synapses that we study. And this is a synapse in the central or peripheral nervous system, stained using acetylcholinesterase histochemistry. Was developed by George Curley more than 50 years ago. So it's of interest is how does the esterase molecule know where it's supposed to go inside the cell, and ultimately what regulates the numbers of molecules that end up at the synapse. So this is sort of a summary of the life history of the acetylcholinesterase molecule. I'm going to be focusing today more on the earliest events of the localized synthesis and transcription, regulation, activation, assembly, and also processing and targeting of the acetylcholinesterase to the various regions of the membrane. Now, this sort of acetylcholinesterase is a membrane glycoprotein. It's synthesized on the rough endoplasmic reticulum like all membrane glycoproteins are. It undergoes, unlike what Christian Anderson thought of most proteins, it's just like all proteins, it undergoes significant modifications. It requires many helper molecules during synthesis and assembly, chaperones. In fact, we know today that there are probably around at least 250 different molecules in the endoplasmic reticulum that help this process along. Acetylcholinesterase is no exception. And this is a picture of the acetylcholinesterase molecule actually from collaboration with Yves Born and Pascal Marchot and PalmerTaylor. And I like the staining, this particular kind of coloration, because it shows the esterase molecule from amino terminus to carboxy terminus, and it gives you an idea of how the molecule might be folding as it's being extruded through the translocon in the endoplasmic reticulum. So acetylcholinesterase starts on the endoplasmic reticulum. The esterase RNA is actually localized on the endoplasmic reticulum. About 50 nucleotides downstream with a stop codon. There's what's called a pumilio binding element, PBE. The pumilio binding element binds an RNA binding protein. It's a repressor, translational repressor. It attaches. There are two pumilio isoforms expressed in mammalian cells. We use antibodies against pumilio 2 that immunoprecipitate acetylcholinesterase messenger RNA measured by PCR. This right here is just a traditional gel shift assay with the pumilio binding portion, the RNA binding portion of the pumilio molecule, showing that it shifts the esterase messenger RNA on a gel. And interestingly enough, pumilio itself is actually bound to the endoplasmic reticulum. This is co-localization of a PUM2 cherry in cells, co-expressed with the GFP labeled SEC61 molecule, and the two are perfectly co-localized. In fact, if we do a crude subcellular fractionation where you have a nuclear fraction, mitochondrial fraction, rough ER, and smooth membranes, the pumilio is actually associated with the rough endoplasmic reticulum fraction of the cells. So the esterase messenger RNA is localized on the endoplasmic reticulum. I should also add, I'm not going to talk about this today, but pumilio, you can think of pumilio as sort of an on-off switch for protein translation. And there are ways of activating it and deactivating it. And probably this is one of the mechanisms for regulating protein translation, specifically at synapses, including acetylcholinesterase. Now, acetylcholinesterase is really unique in a sense. You can actually measure the enzyme, very minuscule quantities of the enzyme because of its really high turnover number. And so you can actually measure the molecules in various subcellular locations very easily. We can treat the cells, and this is one of our standard procedures. It was developed by George Curley over 50 years ago. Treat the cells with an esterase inhibitor, an irreversible one. We use disopropyl fluorophosphate, or DFP. And what that does is it completely inactivates the enzyme in the cell. And then you can look at the synthesis of new molecules as they appear, and then you can treat the cell with protein synthesis inhibitors and chase the catalytically active molecules through the cell. You can measure their accumulation and so on. And you can actually measure their protein processing. So you can think of a conveyor, the esterase being put on a conveyor belt on the endoplasmic reticulum, and that's what we showed here. This is some of my work as a postdoc with Doug Fambro. Cells, these are muscle cells in culture, secrete acetylcholinesterase. The esterase is being produced at a constant rate. You treat the cells with protein synthesis inhibitor here. There's no change in secretion for about 2 hours or so, and then the rate decreases to 0. In other words, the transport time from the ER, from the time it rolls off the ribosome until it gets to the membrane, is about 2 hours, 2.5 hours. This is degradation rate. This is secretion into the medium after DFP, secretion in control cells, secretion in DFP-treated cells. You treat the cell with DFP, there's no appearance in the medium until about 2 to 3 hours, and then it's the same rate. So basically, this is the transport time through the cell. We then also showed that all these parameters were correlated very highly with the transport of the acetylcholine receptor. In fact, this was the first evidence that membrane proteins and secretory proteins were transiting the same pathway in the cell. And I was really happy. These were my 2 first papers as a postdoc. The primary reviewer was George Pilati himself, and that same year after they published, I went to a Gordon conference, and he came up to me and congratulated me. So he thought it was a really nice study, and that really helped me out, in my career. That was a very big word. So the acetylcholinesterase, again, we can measure its processing very easily. For example, we can treat the cells with DFP. We look at the recovery as a function of time, and then we can also look at their binding to different lipids. High mannose residues are added co-translationally, and it turns out all the esterase molecules are co-translationally glycosylated, and the catalytically active molecules appear right away. Then more slowly, N-acetylglucosamine is added on, indicating transport to the Golgi apparatus, the early Golgi apparatus, and it's not until later that you see the addition of more complex sugars, indicating transport to the medial and the late Golgi. In fact, the appearance of the collagen-tailed form of the enzyme only occurs about 1.5 to 2 hours after initial synthesis, suggesting, in fact, that it's actually being assembled in the Golgi apparatus. These studies need to be repeated in other labs, but so far it indicates assembly later on. Now, we thought this was pretty cool. We looked at these molecules very closely. And so now we wanted to study the assembly looking more precisely by using isotopic labeling. We made antibodies to the esterase and began studying using label. And after treatment with DFP, unlike seeing an increase in enzyme activity, we saw a rapid decrease in labeled enzyme, S35-methionine labeled enzyme. In fact, it turns out this was actually the first evidence for the endoplasmic reticulum-associated degradation system. We did the pulse-chase study, and over the first couple of hours, literally 80% of the esterase molecules were being degraded. We actually did a study where we treated the cells with DFP and at the same time labeled them with S35 methionine, and at different times extracted cells and ran them out on sucrose gradients so we could look at the monomeric, dimeric, and this is tetrameric form down here. And what we could see is that at the same time as the S35 methionine labeled esterase molecules were decreasing, there was actually an increase in enzyme activity. So clearly there were two populations of molecules in the cell, catalytically inactive ones that were 80% of the total, and then the catalytically active ones. And it turns out the catalytically inactive ones, for the most part, were monomers and dimers. This is endo-H treatment of esterase forms run out on sucrose gradients, whereas the ones that became catalytically active and were endo-H resistant, these are the ones that went into the Golgi, was really a minority. So clearly there were two populations of esterase molecules, and we wanted to study the ones that were catalytically active a little bit more. And here again, more surprises. The acetylcholinesterase molecule undergoes a very tortuous early development, to say the least. In looking at the esterase molecules that are on the surface of cells, these are tissue-cultured skeletal muscle fibers, and we look at the esterase activity on the cell surface. We have an esterase assay where we can just wash the cells, incubate them with tritiated acetylcholine, and we look at the hydrolysis because acetylcholine doesn't go through the membrane. So we can distinguish between cell surface molecules and those inside the cell. And if we look at cell surface esterase in the presence of, for example, 1 or 5 millimolar dithioethol, there's no change in activity over time. This is based on work that Eric Helenius did to look at the assembly of influenza virus, code proteins and so on. However, if we look at total esterase activity, in the presence of DTT, there's a very rapid decrease. Turns out this is essentially all of this is the intracellular enzyme. The newly synthesized esterase molecules are extremely sensitive to DTT, whereas the ones on the cell surface are incredibly stable. The same thing is true with temperature. And this is based on some studies that Jerry Eichler and Israel Silman did years ago. And we just modified it so we could look at the cell surface enzyme, which is completely stable at 45 degrees centigrade. But the intracellular pool is essentially inactivated quite rapidly, i.e. the esterase molecules that are newly synthesized that are inside the endoplasmic reticulum, in fact, you warm them up, they just fall apart completely. So obviously they're thermally very unstable. They're also very sensitive to proteases. And this just simply shows that in the presence of DTT, it's the monomers and dimers that are falling apart, and in fact the tetramer is completely stable. But even more amazingly, the molecules that actually become converted into active ones is a preset, predetermined pool of esterase. So we did this experiment. It was really quite surprising. If we treat cells with DFP, this is sort of an example of our standard procedure. We DFP treat the cells, we wash it out, and we allow the cells to synthesize new esterase molecules for 60 minutes or 120 minutes. At the end of 60 minutes, in the normal medium, we get this amount of esterase. 120 minutes, we get essentially double that amount. If we allow the cells, treat the cells with DFP, we put them in puromycin-containing medium, you still see a little bit of active enzyme appear, but that's because of a little bit of conversion of inactive to active esterase. And if we allow the cells to synthesize acetylcholinesterase in the presence of dithioethol, there's no enzyme activity. And this is using techniques that were developed by Eric Helenius years ago. Now here, cells were treated, incubated in puromycin for 2 hours, or treated or allowed to incubate and were allowed to recover from DFP in normal medium for 1 hour, followed by puromycin for 1 hour, 1 hour's worth of esterase, of course. And here, the most amazing thing, we allow the cells to synthesize esterase for 60 minutes in the presence of dithioethol. We switch them to puromycin-containing medium, and we get the exact same amount of esterase activity that we did with newly synthesized for 1 hour alone. What this tells us then is that not only do we have inactive esterase being converted to active esterase molecules inside the cell, there's some rate-limiting step, something that's regulating the numbers of molecules that are actually get converted from an inactive to an active state. What could this be? Well, I mean, several possibilities. One could be interaction with molecular chaperones. For example, something might be rate-limiting, protein disulfide isomerase, BIP, any one of the cow-nex and cow-reticulin. There's a laundry list of these molecules. we actually took cells and transfected the cells with several different molecular chaperones. And interestingly enough, we see a fairly large increase in all the higher order oligomeric forms. These are your monomers and dimers separated on sucrose gradients. And this is an increase in acetylcholinesterase. This, I think, is a really important experiment for several reasons. One, it shows that the folding of the enzyme regulated by molecular chaperones can be rate limiting, number one. But also, I think this is a problem that people are really not considering. Everybody assumes that cells, when they're making exportable proteins, the folding itself is not a rate limiting process inside the cell. But clearly, if you can actually increase the numbers of molecular chaperones and you see an increased efficiency of folding, that suggests that, in fact, these glycoproteins in the endoplasmic reticulum are actually competing for the attention of molecular chaperones. And I think this is something that needs to be looked at in greater detail because it really implies that there are levels of regulation here that we don't really understand yet. But not only are molecular chaperones, also the non-catalytic subunits themselves. Here we use the collagen-tailed form as an example. But here, if we incubate cells, just have cells expressing the acetylcholinesterase alone, you get your monomers and dimers. But it's not until you express the collagen tail that you get these higher order oligomeric forms right here. And if you suppress the translation with interfering RNAs, you see a decrease. There's no change in the production of the monomeric and dimeric forms, but there's clearly an increase in active enzyme in the higher order forms. What this tells us then is that the non-catalytic subunits are actually rescuing these catalytic subunits from degradation. These are ones that would normally be degraded, but yet now they're being appeared. And this is going to become quite useful later on, as you'll see, when we talk about therapeutic applications. The other thing, looking at the interactions between the two, molecular chaperones and the non-catalytic subunits, there's actually sort of a synergistic effect. It's more than just simply additive, it's a synergistic effect. And you co-expressing the two, you actually get more than just the sum of the two alone. So there are clearly multiple levels of regulation. So just to sort of summarize then, synthesis of acetylcholinesterase is not just simply a matter of rolling off the ribosome, folding, and boom, here you have this beautiful enzyme molecule. No. A large fraction never see the light of day. They're being rapidly degraded by the ERAD. And those that do survive undergo a period of maybe between 1 and 2 hours where you have this very unstable form. It actually behaves in many respects like a molten globule molecule, but yet it has catalytic activity as well. How this happens, we don't know, and I'm certainly welcome to suggestions. Anything that anybody has any idea on how a molecule can have both of these sets of properties simultaneously is a good question, and I'm looking for suggestions on that. So it goes through this stage where it's highly unstable, and then finally a subset of these molecules become mature, and then end up going onto the cell surface where they're very highly stable. They're completely resistant to proteases, DTT, temperature, and so on. So what happens to the esterase once it's finished folding over here? Now we're interested in how the cell actually targets these molecules. And so one of the types of studies that we're doing right now is looking for targeting of esterase in what are called Maydine-Darby kidney cells, MDCK cells. And this is people who study membrane cell polarity and targeting of proteins to different domains of the cell have used MDCK cells for years. There's certain signaling molecules, signals on esterase on molecules that target them to the apical domain versus the lateral versus the basal domain of cells. And in the case of acetylcholinesterase, you made lentivirus using encoding the different forms of the esterase, the ACHET form that can assemble with either the transmembrane non-catalytic subunit, or called Q. This is the hydrophilic phobic form of the enzyme that's glycolipid anchored. And this is ACHET co-expressed with the collagen tail. And this is just the ACHET alone. And in every case, the esterase molecule is actually targeted towards the apical domain of the cell. In the case of a neuron, this would be equivalent physically to the axon itself. So no matter which esterase form is being expressed, they're all being targeted towards the apical domain of the cell. And just another way of showing that is growing the MDCK cells in these special chambers. This is a filter right here, and you have tissue culture medium on both sides of the cell. We can wash out the tissue culture medium and put in tritiated acetylcholine in each of the compartments, and we start measuring the rate of hydrolysis of esterase in the two compartments. As you can see, in every case, the esterase is being targeted towards the apical domain rather than the basolateral domain. And if we label the esterase with fluorescent fasciculin, snake toxin that binds essentially irreversibly to the esterase, we put it in, it's always on the apical domain, and this is, if it was on the basolateral domain, this is what the staining would look like. So all the esterase is being targeted to the apical domain. This probably tells us that in most cases in the brain, most of the esterase is probably presynaptic in nature. There is a possibility that it's also on the postsynaptic membrane in the dendrites. How would that happen? Well, it turns out that in the case of the PUMIDU binding protein that I was telling you about, this is some unpublished data from a friend's lab, PUMIDU1 gets transported down the axon. PUMIDU2 gets transported out to dendrites. So here's a perfect example where you could, if you really need to retarget a protein, you could actually hook up the RNA to an RNA binding protein, transport it out to dendrites, and then use it as an on-off signal at a synapse to locally translate proteins. So you can see acetylcholinesterase has been a very useful marker for many different biological processes. In the case of muscle cells, we've had papers over the years showing that essentially each nucleus behaves autonomously within the muscle fiber. These are nuclear domains, and they target their locally synthesized proteins to the surface membrane. So if everything is being targeted apically, you can see that at the neuromuscular junction, all the esterase that's being synthesized here will be targeted locally to the overlying region of the plasma membrane. And in fact, that's exactly what happens. In the case of the collagen-tailed esterase, then, once it's made, how does it actually get to the synapse? And this is some experiments that we did in collaboration with Lily Anglister at the Hebrew University. We had postulated that, in fact, these non-catalytic subunits of the esterase are actually acting as retention signals rather than localization signals, if you will. In other words, the esterase is being transported apically, it's dumped out, and then you have signals that retain the molecule. In this case, the collagen-tailed form of the esterase, here we took frozen sections of frog muscle. We have antibodies that recognize avian acetylcholinesterase specifically. It doesn't cross-react with the frog. And we developed a technique where we could purify the collagen tail, or the form that's at the neuromuscular junction, from quail. We produced it. And then we incubated with frozen sections of frog muscle. And what happens is that no esterase, the tetrameric forms of the esterase, but only the collagen-tailed form of the enzyme actually attaches, and it's just sort of a quantitation of this, at the neuromuscular junction. So essentially, they're binding sites at the neuromuscular synapse for the collagen-tailed form of the enzyme. So now it's going to be targeted apically, it gets secreted locally, and now it's bound and retained specifically at the synapse. And I think there are analogous mechanisms for targeting these types of molecules to the cholinergic synapses in the brain as well. But, and this was just some series of studies showing that, in fact, the adhesion site is a molecule called perlecan itself, and there's some mutant mice that were generated in Yamada's lab that are perlecan null. There are also humans with a perlecan mutation where they don't produce perlecan, and they don't have acetylcholinesterase at their neuromuscular junction as well. Okay, so I mentioned the collagen-tailed form and the two non-catalytic subunits. The cDNAs were isolated, encoding these two proteins were isolated in Jean Massouillet's lab a number of years ago by a then postdoc Eric Krejci. And Suzanne Bon and Jean Massouillet studied the structure of the collagen tail, did a series of mapping studies, and they found that this small region right here, 17 amino acids, which they called the proline-rich attachment domain, this is the amino acid sequence right here, actually was the portion that actually attached to the neuromuscular junction. And if you co-expressed constructs containing this region right here, together with acetylcholinesterase, either in oocytes or cell lines, you ended up with oligomerization and secretion of the tetrameric form of the enzyme. In a collaborative study with Israel Silman and Joel Sussman, they produced the peptide. This is a synthetic peptide of the carboxy terminus of acetylcholinesterase, This is the 17 amino acid proline-rich attachment domain peptide, and the crystal structure of the two shows that you end up with parallel alpha helices, and this is the mechanism actually of oligomerization. Well, we looked at this and we said, well, maybe it's possible to do this extracellularly. So we had company GenScript synthesize this peptide for us with a KDL sequence on the end. This is a endoplasmic reticulum localization sequel. This Jim Rothman years ago had shown that if cells take up a peptide with this sequence on the end, the peptide can actually be translocated back to the ER and get glycosylated. So we took this peptide, we have a biotinylated version as well, we incubate cells with it, it gets transported back, and it ends up in the endoplasmic reticulum where it associates with red fluorescent protein, also with a KDL sequence. This is a protein that was given to us by Jennifer Lippincott-Schwartz a number of years ago. So the peptide gets transported back to the ER, and what it does, it induces oligomerization of and produces a large increase in the tetrameric form of the enzyme. So this is the sucrose gradient profiles, the monomeric and dimeric esterase, your tetramer, your collagen tail form, and it's specifically the tetramer that increases quite dramatically. Not only does it increase, we believe that it's actually stabilizing the enzyme, but actually assisting in the folding of the enzyme. It actually assists in the folding, and it helps convert that inactive enzyme. Remember I told you that 80% of the molecules are catalytically inactive. That's partially how we get this large increase in active enzyme. If we treat cells with DFP, total esterase in the cells, treat them with DFP, there's virtually no enzyme activity there. And now we let them synthesize new esterase in the presence or absence of the PRAD peptide. We see that there's a large increase in catalytically active enzyme in the presence of the peptide. But if we look at if we extract the esterase, extract total cell extracts at these different points, and we do a Western blot, there's virtually no change in the total amount of esterase protein. So this tells us that what we're doing is we're actually converting inactive to active esterase molecules. And I also told you that the esterase is being degraded with a very short half-life. In fact, we put in the PRAD peptide. This is a pulse chase study using S35-methionine. It basically rescues most of the esterase and blocks the degradation rate. So now these esterase molecules are going to be exported by the cell. Now, can this be used for anything? And I think the answer is yes. We took the same peptide, the biotinylated version, and we injected it into mice. And these were studies that were done by Susanna Rossi, who's here in the audience today. These are mice that were injected with a biotinylated PRAD peptide, and either 5 or 10 minutes later, dissect out the muscle and look at the distribution of the fixed, and then look at the distribution of the esterase molecules, and you see that there's a lot of endocytosis of the peptide, specifically at the neuromuscular junction. The consequence of this is exactly what happens in the tissue-cultured muscle cells. It produces more acetylcholinesterase. And the way we were able to measure this is by making fluorescent fasciculin. There's a picture of the crystal structure of the fasciculin from Yves Born and Pascal and Palmer. We fluorescent fasciculin, and we use that to quantify or semi-quantify the amount of esterase produced. The fasciculin, there's a lysine up here. We label that lysine with a fluorophore, and the fasciculin binds 1-to-1 with acetylcholinesterase. So the fluorescence binding at the neuromuscular junction is going to be directly proportional to the number of esterase molecules. And what we see is that if we inject mice with the PRAD peptide, this is the very first experiment that we ever did. We injected mice with the peptide, PRAD, and a few hours later, we dissected out the muscle fibers. We ladled them with rhodamine alpha bungarotoxin to measure the fluorescence of the acetylcholine receptors, and fluorescent fasciculin to measure the esterase. And what we see is no change in the levels of receptor fluorescence, but a large increase, in this case 60-some%, in the fluorescence of the acetylcholinesterase. So clearly, even in vivo, when they take up the peptide, it increases the production of acetylcholinesterase, and this new acetylcholinesterase molecules can be inserted specifically into the synapse. I think this is one of the first examples of extraneously modifying protein folding in vivo for a therapeutic application. And we did various time courses. I said we studied this exhaustively. We could do time courses. We can see that even when you see 100% or more increase in the amount of fluorescent acetylcholinesterase, specifically at the synapse, there's no change in the levels of acetylcholinesterase protein in the muscle cell in vivo. And just to show that it's actually functional, we've done this with pre-treatments, post-treatments. This particular experiment, the mice were injected with intramuscularly with a PRAD peptide, and then 24 hours later exposed to a very high dose of DFP, which kills most of the animals. Most of the animals die within 30 minutes or so, but animals that were pre-injected with a PRAD peptide are resistant. So this actually can be used therapeutically, and we now actually have a funded program to use this to treat people who are exposed to nerve agents or pesticides. According to United Nations figures, more than 100,000 people die every year from organophosphate poisoning, and this is a useful way. So basically, the esterase biogenesis, the esterase is synthesized, it undergoes complex folding, and then a lot of this is rescued. And in the next 2 minutes, I want to just have one short study that sort of epitomizes what this symposium is all about, and developing techniques to actually replace inactive esterase molecules. And I kind of like this study because this is actually my study. This is one of the, sometimes I actually go into the lab and play around, and I worked this up from the very beginning, so then I collaborated on this. And given that we could use fluorescent fasciculin to label up acetylcholinesterase molecules, and this is a frog neuromuscular junction in mouse and rat, and so on, why can't we attach other molecules here? So my first experiment was thanks to a wonderful technician who did the molecular biology, Mir Anwar. We took GFP, the green fluorescent protein, we added a leader sequence to it, his tag for purification, a 15 amino acid linker, and then I had GenScript synthesize a humanized version of the fasciculin gene here. So we took this, we expressed this in HEK cells, purified it, incubated it with frozen sections of muscle, and clear enough, it binds to the neuromuscular junction where it co-localizes with acetylcholine receptors. And if you inject it into mice, it does the same thing. Here it goes, it actually co-localizes with the receptors themselves. So if we could do this with GFP, why not do this with acetylcholinesterase itself? So we took the esterase, the mouse acetylcholinesterase molecule that Pascal had played around with, and where they chopped off the last 40 amino acids to make a monomeric form of the esterase. We mutated the 3 amino acids that they used to show that that's where the fasciculin was binding, to make a version of the esterase that was not bound to fasciculin. We have a strip tag here to localize the esterase molecule, and of course we have fasciculin in the linker. This just shows we can purify it. Here we incubate it with fasciculin. This is the wild type, and this is our mutated enzyme. It doesn't bind. And now if we take frozen sections of skeletal muscle fiber, we incubate it with the Karnofsky roots technique. It's a histochemical procedure based on catalytically active enzyme. You can see esterase activity at the neuromuscular junctions. Treat them with DFP, you don't know activity. But then if we incubate it with this construct, we restore enzyme activity at the sites of nerve muscle contact. Can we do this in vivo? And the answer is yes. These are just mice that were injected with our construct, and we could localize it using the strep tag, a special tag on the esterase molecule. And now the question is it functional? And the answer is yes. Mice are treated with an organophosphate, high dose. Most of them die within 30 minutes. But if we inject them a few minutes afterwards with our chimeric construct, we rescue most of them. And this just shows that mice that were treated with DFP and then injected with saline, there's no activity at their neuromuscular junctions, whereas the ones that were injected with the chimeric construct restores enzyme activity. So here we've used the toxin to actually deliver an esterase molecule to a synapse in vivo, and it actually has a therapeutic application. And this can be used for treatment of human. You just substitute the human esterase for the mouse, and this would constitute a viable therapeutic application. So just finally, these are all the people that have worked on this project over the past years, graduate students, postdoc fellows, and of course wonderful collaborators. Thank you very much.
Well, the nice rock punches are waiting, but let's take one question. Way back there.
So, Rich, for lovely talk. I have
on this last thing, you're getting expression in the muscle, right? That's not the nerve, because axon transport would take longer than your time.
That's correct. I mean,
Okay.
Lily Angoster did some studies years ago. Only a very, very small percentage of the estrays at the neuromuscular junction could possibly be delivered by the nerve.
Right. So I wanted to go back to your comments about chaperones and the time for maturation in the ER. Acetylcholinesterase turns out to be a circadianly regulated protein.
It is.
Okay. So does it help you at all, at least for the neuronally expressed, or any of them, if you're time sharing the chaperones by that mechanism? And I don't know the literature, but is that true for the tailed and non-tailed version, that they're circadianly regulated?
That's a good question. That's something, I don't know anybody who studied it. We studied it a little bit of detail. We know that there's an effect of circadian rhythm, which is why we have to design our experiments so that we can take that into consideration. When we do, for example, if we treat cells, treat mice with something and we want to look at them 4 or 6 hours later, we have to have controls at the beginning and the end to compensate, for any changes due to circadian rhythmicity. We know that. But other than that, I don't know of any circadian studies that have been done.
The one paper that I noted says that the cholinesterase message comes up and the production is principally while you're sleeping. That's the leg of the circadian cycle that dominates. And so I just want to put this out there. Maybe this is interesting with regard to cholinergic issues in the aged, because sleep is an issue.
It is. And I looked, and there were some studies that showed that, but I also found studies in the literature where they tried to find circadian rhythms, and there was so much variability that they really couldn't see that much. So it's a good question, and I don't know. In fact, I'm not even sure why you would want to regulate acetylcholinesterase on a circadian, on a circadian basis. Especially like at the neuromuscular junction, Mika Salpeter showed that, for example, the half-life of the esterase is about 25 days. So in many synapses, the turnover is actually fairly slow.
All right. On that note,
Thanks.
I think we should wrap up the session. I'd like to thank Rick and our previous